With radiation, well, I'll talk about chemotherapy first. Chemotherapy, generally what happens is you take this like crazy cocktail of chemicals that identifies any dividing cell inside your body and kills whichever cell is dividing. And so the strategy behind chemotherapy is to kill the cancer faster than it's killing your body, because it really is killing your body. There are lots of cells throughout your body that are just normally and healthily dividing, right? Healthfully dividing. But if you're taking chemotherapy, you're targeting not only those cancerous cells that are dividing, but you're also targeting all of the cells in your GI tract and your skin that are also dividing, and you're killing all of them, which is why individuals who are receiving chemotherapy get so incredibly sick, and why they get incredibly nauseous, right? Because their capacity to digest and process food gets greatly reduced because those cells are all being killed off.
And they lose their hair.
Yep, and they lose their hair. And that's the strategy. You're trying to kill this tumor before the chemotherapy kills your body. And then there's radiation. Radiation can either be a general application or it can be a very targeted application. If you have a localized cancer, like a breast cancer, generally what they'll do is they'll direct a highly radiated beam at that tumor and directly impact that tumor. So when my mother-in-law was going through radiation, they surgically implanted ports on either side, or posts, on either side of the tumor, and then they directed an irradiated beam right at that tumor, and the skin above the tumor got burned really badly, but it killed the tumor. Those are currently the options, yeah.
So I have kind of two questions. So like, if women are pregnant, can they do either of those?
Well, I mean, they could, but the fetus wouldn't survive. It might survive with the directed radiation, but it wouldn't survive with chemotherapy.
And then also, like you always hear people say like they have to have a cure for cancer or something like that. Do you believe that they do have a cure for cancer and they just don't want to give it to people, kind of like a natural selection type of thing, or like?
No, I'm a little bit more cynical than that. I feel like if the pharmaceutical industry could make a ton of money by selling the cure, they would.
Mm-hmm.
I do believe that there is the problem of the haves and the have-nots with pharmaceuticals. So there are treatments that should be available to lots more people than are actually available.
Like in other countries, like
Yeah.
in Europe, they have a great, healthcare is amazing.
Yeah. So socialized medicine is a bad thing to talk about because we have this knee-jerk reaction to it, right? So my father's Canadian, they have socialized medicine, and he was Canadian. I spent most of my summers in Canada. So I'm not as averse to it, but there are costs and benefits associated with socialized medicine. But I mean, if you think about underdeveloped countries and the kinds of things that they contend with, it's heart-wrenching. Okay, so how is it that cancer actually develops? Well, basically what happens is those checkpoints that aren't supposed to be overridden get overridden, and that can happen a couple of ways. Ultimately, it all comes back to some sort of mutation in the DNA. Either our regulatory proteins aren't working correctly because they're manufactured improperly, or something else is going on. But it all leads back to this DNA sort of mutation idea. There are two different types of cancerous genes that we need to talk about. The first of which are called proto-oncogenes, and the second of which are tumor suppressor genes. So proto-oncogenes, these are genes that actually stimulate the cell cycle, right? So these are the growth factors, basically. And if there's some sort of mutation in the regions of the DNA that are regulating the production of growth factors, then we can have serious problems. So an interesting thing about DNA is most of our DNA doesn't code for anything. It's just kind of space, gap junk DNA. But genes live in regulatory regions. And so generally upstream from a gene, there is some sort of regulatory gene that's dictating whether or not the downstream gene is going to be replicated or produced, that gene product will be produced. If there is a mutation in that regulatory region, what can happen is that gene product that's coded downstream from it can be overproduced. And so if we start overproducing our growth factors, then cells can start going through uncontrolled cell growth. If that happens, if a mutation occurs in a proto-oncogene, it becomes what's called an oncogene or a cancer gene. So that's way number one, overproduction of growth factors. Secondly, there are tumor suppressor genes. They're genes that actually produce proteins that go out and look for cancerous cells and tumors. When they find them, they initiate apoptosis and those cancers are taken care of. If, however, a mutation occurs in a tumor suppressor gene region, then we no longer have that regulatory mechanism, and cancerous tumors can grow at their will and pleasure. Okay, does that make sense? So two ways that we can get cancers, one of which is overproduction of growth factors from mutations in the proto-oncogenes. Second of which is a mutation in the tumor suppressor gene region, where we stop that tumor suppression protein production. And this is kind of how that looks. So there is a protein, and what I hope you notice here is I've written P53 two different ways. So P53 non-italicized and P53 italicized. If you see that, what that means is P53 italicized refers to the actual gene, and the non-italicized refers to the protein that it's making. So there is a gene region called P53, and it produces the P53 protein. And that P53 protein is kind of a regulatory protein. It allows us to go past the G1 region. If it's working healthy, what happens is our DNA begins to grow and to divide, right, and we replicate our DNA, and then we hit the G1 checkpoint. P53 is initiated. And then if there's a problem in the DNA, like if there's a problem in this region right here, P53 initiates an enzymatic correction. An enzyme comes out and it looks at the DNA and it tries to correct it. If it corrects it, then that cell can continue to go through the cell cycle. If that enzyme can't correct it, then apoptosis is initiated and the cell is digested. If, however, there is a mutation in the P53 region, then we can't produce that P53 protein correctly. So if there's a problem in our DNA, we can't regulate that, we can't fix it, we can't assess it. And that cell will just continue to go through the cell cycle. And we'll get some healthy cells produced, but we'll also get some cancerous cells produced. Okay.
Can you explain which italicized and non-italicized?
Sure. Non-italicized is the protein product, and italicized is the gene region. Unfortunately, we name them the same thing. Other questions? Okay. So in terms of chapter 8, right, this is the chapter 1. In terms of the mitosis chapter, I want you to be familiar with the cell cycle, those three phases, right? Interphase, mitosis, cytokinesis. I want you to be familiar with the phases within mitosis and what happens in each of those, prophase, metaphase, anaphase, and telophase. I want you to be able to talk about the difference between cytokinesis in animal cells and plant cells. The cell cycle regulation, so talk about the checkpoints, be familiar with the checkpoints, and then cancer and cancer regulation. Okay? Questions about any of those things?
Can you repeat that whole thing?
That whole thing?
Well, just like from, just like what you want us to know. From the cell, I got the cell cycle, 3 phases, the process within the cell cycle.
So mitosis,
Yeah,
the phases of mitosis, know what happens in each phase of mitosis.
okay.
Know the difference between animal and plant cytokinesis. Cell cycle regulation, the checkpoint system. Cancer, know the difference between like malignant and benign tumors, and then cancer regulation.
Thank you.
You bet. Other questions? Okay. Sorry, let me